Seasonal changes can also affect the phenotype, both for animals and even for plants. So think of the trees that lose their leaves in the winter. That's a change to their phenotype that's brought about by seasonal change. There are many animals that have seasonal changes in their phenotype, like this Arctic fox, who in the winter has a white coat and in the summer has a brown coat. Its genotype remains the same. Only the phenotype changes in response to usually changing day length changes the phenotype. Another multifactorial trait that I mentioned before was IQ. And IQ is probably one of the most heavily studied traits in psychology. So we have a lot of data on the effects of genetics versus environment on IQ. Although IQ testing is controversial and it's very difficult to design a test that doesn't have some sort of bias. But however you're testing it, because we do get a bell curve of distribution for whatever scores you're getting, that indicates that it is most likely polygenic. And the fact that it changes with changing environments indicates that it's also multifactorial, meaning that there's environment and a genetics component to it. Twin studies is one of the ways that we study in humans whether a trait is completely genetic, completely due to the environment, or is a mixture of the two and is a multifactorial. I put in our playlist a video that explains this very well, how we use twin studies to tell whether something has an environmental as well as a genetic component by studying twins, identical twins versus fraternal twins, identical twins versus siblings in the same family that are not twins, and how that can tell us whether something has an environmental component and how much of an environmental component there is. So for traits that are multifactorial, we give them a heritability score, which is a percentage. So a heritability score of 1 would mean that it's 100% due to your alleles and 0% due to the environment. If it's a heritability of 0, then that means it has no genetic component whatsoever and it's completely due to the environment. So heritability for IQ, depending on the test and the situation, is like 50 to 70% is the heritability of IQ. So that indicates it does have a strong genetic component, but it is also heavily affected by environment. And obviously nutrition is going to play a big part in that. That's part of the environment. So very poor nutrition, and by poor nutrition I don't mean people who eat at McDonald's a lot or something. That's not what I mean by poor nutrition. I mean inadequate nutrition, vitamin deficiency, iron deficiency, protein deficiency. These things can lower, and just lack of calories. These things can lower IQ. Level of education and where that education is taking place also have an effect on IQ. As you go through school, that affects your IQ. We also know stress affects IQ as well, negatively. And this is a chronic long-term thing with children, especially who are under chronic stress. This can lower their IQ, and it appears to be partially recoverable as well if the stress is removed. And this would be severe stress, things like being homeless, having a scary, violent adult living in the household with them. That's the kind of stress that I mean. There was an old hypothesis that was based on rat experiments in the 1960s that evolved into the enriching environment hypothesis. And this was the rat experiments, which you'll hear about if you're going to take Intro to Psych. And what they did was they put rats in empty cages versus rats that had toys. And the rats that had toys in their cages did better finding their way through mazes than rats in empty cages. And so the hypothesis was the equivalent of that for humans would be, giving kids lots of books and toys would raise their IQ, and that didn't happen. And then somebody did another experiment with rats where they gave them not toys, but friends. So they had other rats in their cage, and those rats did even better. So, and rats are social, so are humans. So an enriching environment for a human is not an environment full of toys and books. An enriching environment is one in which there's lots of interaction with other humans. But this is a very enormous topic in psychology. So if you're going to take Intro to Psych, you're certainly going to hear about this topic. So we talked a bit before when we talked about evolution about mutations, and that mutations that are going to lead to evolution are ones that are available to natural selection. So now that we have defined more fully what a phenotype versus genotype is, so a mutation that does not change the phenotype is not available for natural selection to act on. Natural selection acts on phenotypes, whether those phenotypes are physical traits or behaviors. That's what natural selection is acting on. So if a mutation changes the DNA sequence, but it's not in a gene, it's between genes, and it has no effect on it, it's invisible to natural selection. So here's some of the terms that we've talked about so far. And so make sure that you can differentiate between these terms, genes versus alleles. So alleles are different versions of a gene. Homozygous means an individual has two of the same allele for a particular gene. Heterozygous means they have two different alleles. Genotype is which alleles you have. Phenotype is the expression of those alleles. So what do we see? What's visible? What's measurable about that organism? That's the phenotype. We talked about dominant versus recessive traits, and Mendelian traits have a complete dominance in their expression, but we also talked about incomplete dominance and codominance. So incomplete dominance means that one allele is not completely dominant over the other one, and you can tell who the heterozygotes are. There's a third phenotype. Codominance, it's very hard to come up with good examples for that. The best one is the A and B blood groups. We talked about independent assortment of traits. Now this is due to, this is a phrase that we used in meiosis to talk about how the chromosomes separate independently. The maternal and paternal versions of the chromosomes go randomly to one of the daughter cells during meiosis, and Mendel found the same thing for his traits, and that's because almost all of the traits that he picked to look at in peas happen to be on different chromosomes. Polygenic traits, poly means many, so polygenic means a trait that's controlled by several or many genes, like height or IQ or skin color. These are controlled by several genes, not just a single gene. If we say single gene trait, that means there's just one gene controlling that particular trait. We talked a bit about pleiotropic effects. So pleiotropic effects is becoming an old-fashioned term because now we know the molecular basis of many, many traits. But it's used in genetics to describe one gene that seems to have an effect on different things in an organism that seem to be unrelated. That's pleiotropic. So we are, of course, really interested in human genetics, although doing experiments in human genetics is not acceptable for many reasons. One reason is because human generation time is very long and parents have very few offspring, and also just doing experiments with humans is morally unacceptable. But we can still study human genetics, and we do it by looking at family trees and creating what we call a pedigree for different families, and we can look at traits and tell whether traits are behaving like Mendelian traits. Is there a dominant and a recessive, or does it seem to be incomplete dominance or something else? And so we'll do some problems with those. So a pedigree is a family tree, and the way we construct them, there's particular rules for making a pedigree. And you always have the older generations are at the top, younger generations are at the bottom. If there's a lot of generations, we will number them with Roman numerals. And this allows us to talk about specific individuals more easily. We can say, oh, you know, look at the males in generation 3, for example, and then we would know, oh, we're going to look along this line here. So, and generally, Squares are males, circles are females. A horizontal line connecting two people indicates a couple that's going to have children. A vertical line is connecting to the children. So this couple here has only 2 children. So this female here is a child, and this male here is one of their children. These other 2 in that generation 2 are connected by a horizontal line, so that indicates spouses here. To those individuals. And then we have a vertical line again connecting them to their children. So that's how you tell who's a blood relative in a family tree and who's not. And generally we'll have some kind of a key explaining what the dark and the light ones mean. So if we're interested in a particular disorder or some particular trait like eye color, so maybe we would say, okay, the dark ones here, those are individuals with brown eyes. The light ones there, those are individuals with blue or green eyes, and then that would explain the tree. So usually that's how we do it for a pedigree. Usually whatever the trait is that we're specifically interested in, that's going to be the ones that are colored in there. But there'll always be a key explaining that. So, but using pedigree analysis like this, it helps us figure out if human traits are Mendelian traits. Are they probably polygenic traits? And we'll be doing some examples of these. Okay, so here we have 2 traits that in humans behave like Mendelian traits. So there's a definite dominant and recessive. One of them is having a widow's peak. So a widow's peak is a little point in your hair in the front. I have a widow's peak. And widow's peak is dominant, and it's a Mendelian dominant. So that means that if you don't have a widow's peak, if your hair goes straight across, now if you have a receding hairline, it might be hard to tell. You might have to look at some pictures of yourself from when you were a kid to be sure whether or not you have a widow's peak. If your hair goes straight across, that's the recessive phenotype. So for somebody expressing the recessive phenotype, we know their genotype. They have to be homozygous for the recessive allele if they're expressing the recessive phenotype. So if you don't have a widow's peak, then we know your genotype. If you have a widow's peak, we don't know right away what your genotype is because there's 2 possibilities. If you have the dominant phenotype, you might be homozygous or heterozygous. We have to look at the rest of the family tree to tell. So, okay, so here's our little pedigree here. So the colored in individuals here have a widow's peak, and the open circles and squares do not have a widow's peak. So that's what affected means. So in this case, the trait is widow's peak. So affected means they have the widow's peak. Okay, so if we look here, we see, okay, so for some of these individuals, we can definitely figure out what their genotype is. So in this case, this couple here, one spouse doesn't have it. So she only has recessive alleles to give to the next generation. So in that case, we know every child of this couple is going to have at least one recessive allele. So we know that the 2 individuals here expressing the dominant trait, they have to be heterozygous because they got their dominant allele from the dad here, and they had to get a recessive allele from the mom because she's only got recessive alleles. And children get 1 allele from each parent. One was in the egg, one was in the sperm. So these individuals have to be heterozygous. And here we see that same thing again. 1 unaffected parent with the recessive phenotype. So that means every child has to have at least 1 recessive allele. So we know this individual here is heterozygous as well. So we have 2 heterozygous parents here, but this individual, we don't know. We don't know whether she is homozygous or heterozygous because she doesn't have any kids, and because both parents are heterozygous, she could be either. We don't know. Somebody who's recessive, though, we know. So, and this is something that we see for a dominant trait. So if you're looking at a pedigree and you want to know, is this trait a dominant trait, which widow's peak is a dominant trait. And so the way that you tell is you start at the bottom, look at the kids. Every affected child has to have an affected parent. Child, parent, child, parent, child, parent. Every affected child has an affected parent. Because remember, for a dominant trait, They cannot skip a generation. Dominant alleles cannot skip a generation. We always see them. If the dominant trait is there, we're going to see it in the phenotype. So widow's peak is dominant. It does not skip a generation. Now over here is a recessive trait. Attached earlobe. So free earlobe is more common, but just because it's more common, that does not necessarily mean that it's dominant. It just happens to be. So free earlobe is dominant, attached earlobe is recessive. So attached earlobe means that you don't have a space between your earlobe and your head. It goes straight across. That's attached earlobe. So that's recessive. And this also behaves like a Mendelian trait. So in this case, it's the recessive individuals that are purple because that's the trait we're interested in. We're interested in attached earlobe. So those are the individuals that are colored in. And now here we can see something that we look for in a pedigree if a trait is recessive, and that is skipped generation. So here we have affected child, unaffected parents, affected child, unaffected parents. And that is something you do not see with the dominant trait. Over here, where the trait is dominant, we see child, affected parent, affected parent, affected child, affected parent. You don't see that skip generation, but here we have unaffected parents, affected child, unaffected parents, affected child. And that is definitive for a recessive trait. That tells us this trait is recessive because it skipped this generation, skipped this generation. So we can tell that that is recessive trait. That's what we're looking for a recessive trait. Now, new mutations are interesting to geneticists, as are traits that are very common in one particular family. So if there's a trait that's rare generally in a population and really common in one particular family, that is of interest. And this is a different case. This was a new mutation that appeared. So this is a woman who lived she was born in the late 1890s, I think, if I remember the story correctly. And she had what they called later the Norwegian woolly hair gene. And nobody in her family had curly hair, but her hair wasn't just curly. Her hair would snap off. So if she rubbed her head, her hair would just break off. So very brittle hair as well. And so you can see she has very short hair because her hair is very brittle and just breaks off. And so here's her family pedigree down here. So she's right here. She's this colored in individual here. So all the affected individuals in her family, so we have 5 generations of her family here, all the affected individuals are colored in dark. And the white circles and squares are the unaffected individuals. So now there was no DNA sequencing or figuring out what the gene was when this was being studied in the 20s, 30s, 40s, and this tree was being developed. But we can tell from this tree whether this allele is dominant or recessive, and also whether it's a sex-linked trait or an autosomal trait. So autosomal, this is referring to the autosomal chromosome. So remember, we have 22 pairs of autosomal chromosomes and 1 pair of sex chromosomes. So an autosomal trait is on one of those 22 autosomal chromosomes, which everybody has 2 of those. If it's a sex-linked trait, then that means usually it means it's on the X chromosome. Occasionally it might be on the Y chromosome. But in that case, we're going to see very different distribution. Generally, that's going to mean we're going to see it mostly in boys. So if we look here, and we'll talk more about sex-linked and X-linked traits later. So if we look at this tree, if we want to know first, dominant or recessive. Okay, so we want to know, does it skip a generation? Do we see unaffected parents, affected child? So if we go look down here, affected child, parent, child, parent, child, parent, child, parent. So every place that you look, affected child, there is an affected parent. And that is what we're looking for dominant. So this, in fact, is a dominant trait. Every place you see unaffected parents, no affected children. Unaffected parents, no affected children. Unaffected parents, no affected children. So this is a dominant trait, which also tells us that this is not a null allele. A null allele is going to be a recessive allele. So that means this gene is doing something. It's mutated. It's mutated, but it's still being transcribed. It's still making a gene product. Recessive alleles are important in human genetics because so many recessive genetic disorders are very rare, and it can often be difficult to identify carriers. So a carrier is somebody who is heterozygous for some recessive allele, and usually we're only talking about, when we say someone's a carrier, we're referring to a deleterious allele, something that causes some sort of genetic disorder. Normally we wouldn't use the word carrier if we're talking about somebody who's heterozygous for blue eyes, for example. So carrier means they don't have the condition, whatever it is. If it's a recessive genetic condition, then that means you have to be homozygous for the recessive allele and have 2 copies of it in order to express it in your phenotype. So carriers are phenotypically normal. We cannot identify them as carriers based on their phenotype. And this is, I mean, there are thousands of recessive genetic disorders. And that's because, if you remember, recessive genetic disorders are often null alleles. And that a mutation is much more likely to make it turn an allele into a null allele than it is to make it into an active but malfunctioning allele. So this shows a little family pedigree here of what we talked about before, of what we typically see for a recessive disorder, and that is a skip generation here. Mom and dad unaffected, phenotypically normal, 1 child who is affected, and that is what we are looking for to identify something as a recessive condition. If this was a dominant condition, if whatever this condition is was caused by a dominant allele, one of the parents would also have to have the phenotype. So be careful during the exam if the question is talking about someone being a carrier or being affected. So if we say somebody is affected by cystic fibrosis, that means they have it. It's in their phenotype. If somebody is a carrier for cystic fibrosis, that means they are unaffected, they don't have it, they're phenotypically normal, but they have the allele. They're carrying the allele. Think of it that way. Sort of like carrying it. You're carrying it in a bag, but you're not using it. That's what being a carrier is for a recessive disorder. So here's an example for cystic fibrosis of what we see when we do a Punnett square. So in order to be affected, cystic fibrosis is recessive. So this is the one that we talked about before, that is caused by a defect in a particular membrane transport protein. This transport protein is found in mucous membranes throughout the body, primarily in the lungs, but also in liver and pancreatic ducts and in the intestines, any place you have mucus secretion. And so individuals who are affected, that means they're homozygous for this recessive allele. And individuals who are homozygous for the recessive allele are going to have the condition in their phenotype. So in order to get 2 alleles, that means one has to come from each parent. So you'll notice here, so the capital F here designates the dominant normal gene for the protein, the cystic fibrosis protein. The lowercase is the recessive allele that leads to cystic fibrosis. So if we have an affected child, both parents have to be affected or have to be carriers, because that's the only way you can get a child who is homozygous. Now, it is possible that there could be a new mutation, and very rarely we might have one parent who's a carrier and the other parent is not a carrier, but there's a new mutation and we could end up with a child who's affected. That's far, far less likely, though, than the possibility that both parents are carriers. So if we have an affected child, generally we don't even have to test the parents, because we just assume that both parents are carriers for this. And so we can see here from the Punnett square that this means that of any pregnancy for this couple, that particular pregnancy, there is a 25% chance that that child will be affected. There is a 50% chance that the child is going to be a carrier like the parents, and there's a 25% chance that the child will be not even a carrier and not inherit the cystic fibrosis allele at all. And this is true for any recessive genetic disorder. We would see a similar pattern like this. And because almost all of these recessive alleles are very rare in the human population, we see very, very few affected individuals, 1 in tens of thousands generally for many of these recessive genetic disorders. Now, there's a couple of recessive genetic disorders that are not rare in particular populations, and cystic fibrosis is one of those. So it's of interest specifically because, unlike almost every other genetic disorder, cystic fibrosis is not as rare as we would think it is for something that until extremely recently in human history was 100% lethal in childhood. So there is going to be strong natural selective pressure against this allele because prior to even until the 1970s, individuals with cystic fibrosis rarely made it to adulthood, and so that means that they did not reproduce and pass on their alleles. So that's a strong negative selective force in that case. So when we see that, and then we see that the allele itself is not that rare in particular populations, that leads us to question, is there another selective force going on here? And so we're going to talk about that because, and we'll talk also about sickle cell disease, which is another one that is, although until very recently it was 100% lethal in childhood, that's a strong negative selective force. It's not that rare in certain populations. So why is that? These questions are very interesting to geneticists and also evolutionary biologists. So here's another recessive condition that is not nearly as deleterious, and that is albinism. Albinism occurs throughout the entire human population, although it's rare. And so here we have on the right here's a little girl who has albinism and her normally pigmented sibling. And there are several different kinds of albinism. There's a number of genes that can be affected. So in order for an individual to have albinism, they have to have a recessive allele, 2 recessive alleles at the same gene site. So there are multiple enzymes that contribute to the synthesis of melanin, the pigment that's in our skin. And it doesn't matter if your skin is fairly light or dark, it's the same melanin, unless you're a redhead, then you have a modified melanin. But for the rest of us, it's the same melanin, it's just the amount that you have. So for individuals who are albino, either they don't make any pigment whatsoever, and the defect is in one of the enzymes in the melanin synthesis pathway, or they make melanin and it's just not deposited in the skin. It's not made by the skin melanocytes, but it might still be present in other organs like the pineal gland and the retina of the eye, non-skin tissue. So there are multiple kinds of albinism, and it's extremely rare throughout the human population. So that tells us that it's deleterious enough to be selected against. So individuals who are albino have varying health problems based on what kind of albinism they have, but the biggest problem that you might guess is they have no protection from the sun. Because melanin, one of its functions is to protect our skin from damage from the sun. But you notice here, just like for cystic fibrosis, both parents have to be carriers, and they have to be carriers specifically of a recessive allele at the same gene. So even though there's 8 or so genes that can be affected by albinism, if they have a recessive allele for 2 different genes, they will not have the phenotype. And in fact, they'll be a carrier for 2 different types of albinism. So when there is a family or a population that a normally rare disease-causing allele is common, that becomes really interesting to geneticists, especially because it can help us identify the gene involved. So this is a particular small population of Native American Hopis who I believe they were living in New Mexico. It was discovered that by some geneticists that they had a very high occurrence of albinism. And so here is so that makes it interesting. It's much more interesting to study a rare genetic allele if you happen to have a family where there's a lot of people who have it. Okay, so here we see what we would typically see for a recessive allele, and the carriers here who have the half circles and squares, they've been identified not by some kind of DNA analysis, but by just looking at the family tree. So let's look at how those were figured out. So the affected individuals are colored in, and that's what we typically do in a pedigree. Whatever trait we're interested in, the affected individuals are colored in. Oh, now I was going to tell you about this picture. So this is a picture from, I believe it was the 1930s, of three sisters here, and one of them is affected by albinism, as you can see. But if we look here, so the affected individuals are colored in, and you can see for this one extended family here, there's a lot of individuals who are affected with albinism, and that's why this particular group of families was interesting to geneticists. So what we can tell here, so here's an affected individual. Both their parents have to be carriers. So if we have a child born with a recessive genetic condition, that means the parents are both carriers. So that's why they are colored in as half circles and squares. The same here. Child is affected, so both parents are very likely carriers. Now some of the individuals, we don't know if they're carriers or not. We just know that they're unaffected. So if they had no children of their own, like these individuals here, or at least their children aren't indicated on this pedigree, then we can't be sure whether they are carriers or not. But, you know, children of carriers have a 50% chance of being carriers. So there's a pretty good chance that these individuals here are carriers. We just don't know based on the information that we have. Okay, so now let's look here. So here we have somebody who is affected, who has a spouse who is not affected. And then we look at their children. Now, this is a good indication that this spouse here, the mom, was not even heterozygous. She's not a carrier because all three children are phenotypically normal. But we also know they're all going to be carriers because they have an affected parent. So if a parent, and this is true even if it's not a disease-causing recessive allele, if it's something like blue eyes, that if one of your parents has blue eyes and you have brown eyes, you have the blue eye allele because that's the only allele that you could have gotten from your blue-eyed parent. So same here. So one parent is homozygous for the albinism allele, and we know that because they're affected. All the children have to have at least one copy of that allele because that's the only allele they could get from that parent. And if you remember from meiosis, those two alleles separate during gamete formation, and 50% of the offspring get one and 50% of the offspring get the other. Well, if they're both the same, if that parent is homozygous, that means all the offspring are going to get that allele, at least one copy of it. And if they don't get it from the other parent, then they're not going to be affected, but they'll all be carriers. And here down here we see something interesting that we also see with recessive traits, and that is that if both parents are homozygous and have the trait, all the children are going to have it. So think of red hair in humans. So think of if you've ever watched any of the Harry Potters, the Weasley family in Harry Potter, both parents have red hair, all the kids have red hair. And that's what we expect to see for a recessive genetic disorder because the parents only have recessive alleles. What else are the kids going to get? So all of the children affected as well. So if both parents have albinism, then all the children are going to have albinism as well, as long as it's the same allele. If the parents had two different types of albinism, then none of the children would have albinism because if they were affecting different genes. The children would all be heterozygous for both genes. So now if we also look here, now we haven't started talking about sex-linked traits specifically, but we will in the next section. So, and again, mostly what we're talking about when we say a trait is sex-linked is that it's a gene on the X chromosome, and we'll talk about why that is. But we see if it was a recessive genetic disorder that was a sex-linked trait, that would mean we would mostly see it in boys. And in this family tree, we see quite a few affected girls. So no, so it's not a sex-linked trait, so that means it's this gene, whichever gene this particular kind of albinism is, that means that it's on one of the 22 pairs of autosomal chromosomes. So that's what autosomal means. So if we say something is a recessive autosomal disorder, that means that it's on one of the 22 autosomal chromosomes. So we already talked about this as recessive. If this was a dominant trait, then we would see every time there was an affected child, they would have an affected parent. That's what we expect to see when a trait is dominant. So this clearly is not that. So because almost all recessive genetic disorders are very rare, the chance that by just random mating we're going to get two people who are carriers of the same recessive genetic disorder are going to meet and have children together is very low, and that's why almost all of these deleterious recessive alleles are in fact very rare in the population, and the disorders they cause are very rare. The exception to that is when, well, we'll talk about another exception that has to do with natural selection, but the biggest exception is if you have close relatives mating. So we call those consanguineous matings, and you can see the root in here for blood. Sanguine and con is with, so consanguineous matings are people who are blood relatives, and generally we mean close blood relatives. So how do we know how close a relative is too close? Well, if you consider that there's estimates that each individual human has approximately 10 recessive genetic disorder alleles. So out of your 25,000 genes that you have in your genome, about 10 of them are alleles. You have, you're heterozygous, unless you have a recessive genetic disorder, you're heterozygous for 10 different recessive genetic disorders. But the question is, what are the chances that the person you're going to have children with has even one of those in common with you? And the chances are, if you're just randomly meeting people who aren't blood relatives, the chances are very low. However, if you married your first cousin, or a half sibling, or a full sibling, the chances that the two of you have the same recessive genetic disorders goes up dramatically. So you share 50% of your DNA with siblings. So let's look at this little chart here to try to figure out how close a relative becomes risky for having recessive genetic disorders appear. So self is here. So you share about 50% of your parents' DNA. So you have about, because you got one of your mother's of each of her chromosomes, and one of each of your father's pairs of homologous chromosomes as well. So you share 50% of your DNA with your parents, and also with your children. You share 50% of your DNA with your children. Full siblings, so a full sibling, that means you share both your mother and your father are the same. You share 50% of your alleles with a full sibling. So if you have 10 recessive genetic disease alleles and your sibling has 10 genetic disease-causing alleles, chances are you share 5 of those with your sibling. So if two full siblings have children together, the chances are really good that they're going to have a child that has a recessive genetic disorder. With nieces and nephews, you share about 1/4 of your DNA. So that's the next degree. Half siblings as well, about 1/4. Your grandparents, you also share about 1/4. So that would mean there's still a really good chance that somebody who shares who's that close to you, half siblings, nieces or nephews, chances are pretty good that you have 2 or 3 of the same recessive genetic disease-causing alleles between them. So somebody who married somebody who shared 1/4 of their DNA, also chances are pretty good that they're going to have a child who has a recessive genetic disorder. First cousins. So first cousins, you share your grandparents. You have one set of grandparents in common, and you share 1/8 of your DNA with first cousins. So chances are still pretty good that at least one of the recessive genetic disease-causing alleles that you have is in common with your cousin. If you share 1/8 of your DNA and you have about 10 disease-causing alleles, then chances are pretty good that a first cousin is going to share that. And that's probably why almost everywhere in the US and most of the world as well, it is illegal to marry a first cousin or closer. So it's illegal to marry a first cousin, a half sibling, a niece or nephew, aunt or uncle, your grandparents, or your parents. That's against the law. Those are all too close relatives. Now, if we go to second cousins now, so second cousins would be would mean that your parents and their parents are first cousins. That's second cousins. So second cousins share great-grandparents. So that means you have one set of great-grandparents in common. And that's 2 more degrees of separation. So you to your second cousins, instead of 1/8, you only share 1/32 of your DNA with them, or about 0.04 or 0.03% of your DNA. So now we're getting to the point where the chances that you and your second cousin share the same recessive disease-causing alleles are less than 50%. So that's generally where most states draw the line and say, okay, you can't marry your first cousin, but you could marry a second cousin, and many people do. In fact, one of our presidents, Roosevelt, FDR, was married to a second or a third cousin. I can't remember. Eleanor Roosevelt was already named Roosevelt when she married Franklin. There are states where it is legal to marry your first cousin. I will let you guess which states those are. Okay, so recessive genetic disorders that are not rare are interesting to both geneticists and evolutionary biologists, because we wonder if there's an evolutionary reason why these recessive alleles that seem to be nothing but deleterious are common in particular populations. So let's talk about sickle cell disorder. So in sickle cell, individuals have a single nucleotide change in the beta globin gene. And beta globin is one of 2 globins that form hemoglobin, which has alpha globin and beta globin. And this one change turns a hydrophilic amino acid side chain into a hydrophobic one. And hydrophobic molecules stick together in a watery environment, like oil droplets sticking together in salad dressing. So the hydrophobic regions of the mutated beta globin attract each other and stick together into these long strands, and that's why you get the sickle shape of the red blood cells. They rupture prematurely, and that's what causes all of the problems for individuals who have this. So you get clotting, pain. It's an extremely unpleasant disorder to have, and without medical treatment, it is fatal in childhood. Even with really good medical care now, it still it results in hospitalizations and a lot of sick time for individuals who have it. So until the last 100 years, individuals who had this disorder did not reproduce. So that's a really strong negative selection pressure on this allele, which should make this allele rare, but it's not. So why not? So, before we go on to that, let's just look at our little family tree down here, where we can clearly see that this allele is recessive. So here is an affected individual here. And so all of the children are going to be carriers because she only has recessive alleles. And then there's one single grandchild down here who's affected, who had 2 parents who were carriers. So the question, why is it common? So why is that even a question? Almost every recessive genetic disorder is going to be very, very rare because all we have is the negative selective pressure on individuals who have this disorder. So here we have, so the homozygous individuals, we'll call that negative selection pressure. Individuals who are phenotypically normal, we have positive selection for that. So what we sometimes suspect is happening when an allele that's deleterious is common is that in fact heterozygotes have an enhanced survivability in some particular environment. So I'm going to put lots of positive arrows here for that. So if that's the case, if for a particular allele, having a double dose of it, as in the homozygous case here, is fatal, but having a single dose of it is highly beneficial, then we will get this deleterious allele being way more common in a population than we think it should be. And we call that heterozygote advantage. So somebody noticed many decades ago that the area where the sickle cell allele is the most common overlaps almost exactly where we find the most deadly type of malaria, or I should say the only deadly type of malaria. Malaria is caused by a protist parasite, so it's a eukaryotic parasite that is transmitted by mosquitoes, which probably most people know. There are 7 species of the malaria parasite that can infect humans. Only one of them causes fatal infections, and that's the falciparum species of malaria. So where we find this in Africa, that's where we find the sickle cell allele. So the green here is the distribution of sickle cell allele in Africa. And you can see that in some areas, the percentage of individuals who are carriers of this is close to 20% of the population. So, and those are, coincidentally, areas where we also find this deadly form of malaria. Now you might wonder, what's happening up here? Why is sickle cell allele not common here, where we also, and over here, where we also have the deadly form of malaria? Well, a different allele is common there, and I'll talk about that. So this is what we call heterozygote advantage. And what turned out is that if you are heterozygous for the sickle cell allele, you are more likely to survive a childhood bout with malaria than somebody who's homozygous for the normal beta globin allele. And that's because of the way this parasite reproduces in the human body. It requires red blood cells that last for weeks and weeks. And it turns out that heterozygotes, people who are carriers for sickle cell, their red blood cells don't last quite as long as people who are homozygous for the normal beta globin allele. So that means that the malaria parasite has trouble reproducing in people who are heterozygous for the sickle cell allele. So that means that they're going to still get it, but they're not going to get as sick, and they're going to tend to survive a childhood infection with malaria. So we have this huge positive selective advantage in these areas. So now if this is true from a natural selection point of view, that heterozygotes have an advantage only in places where there's malaria, well, what about people who whose ancestors are from this area, but who no longer live in malaria areas, like African Americans? If this is true, we haven't had malaria in the United States for over 100 years. So if this is true, we should be seeing a decrease slowly, generation after generation, in the prevalence of the sickle cell allele. And in fact, that has happened among African Americans, that the prevalence, the absolute prevalence of the sickle cell allele has decreased a bit in the last 100 years. Now remember, natural selection occurs in populations over generations, so it's a slow process, but that is what's happening, and that is exactly what we would predict if the reason for the high prevalence of the sickle cell allele is in fact an evolutionary one. If you take away the selection, if you take away the positive selection pressure put on heterozygotes by the malaria parasite, that allele no longer has any advantage at all, and in fact all we're left with is the negative selection pressure on homozygous individuals who have that. So that's what we see. Now, malaria is present in a lot more areas in the world other than Africa, and in fact other mutant hemoglobin alleles have evolved to protect against malaria by heterozygote advantage. We have thalassemia, and there's both beta thalassemia and a so-called alpha thalassemia, and those are present everywhere that you see orange here. So sickle cell here, prevalence is in fuchsia, and you can see a few little pockets of it here, which I have interesting stories behind them. The little pocket of sickle cell that's present here in India is due to the fact that there was some ancient Indian ruler who liked to have African guards, and they imported a lot of guards from Africa during trade. So we end up with a little pocket of sickle cell incidents in India. And there are several other more rare variants here represented by the green and the blue colors here. Some of them are quite interesting and are less deadly, like this little one that's represented here by the blue color in Western India. Some of them are less deadly than sickle cell in the homozygous form, but still provide heterozygote advantage to carriers. So we can see that this isn't just a singular event, that mutant forms of globin genes have been selected for to help protect the carriers against malaria. And this is natural selection in action. Another common deleterious allele is the cystic fibrosis allele, which we talked a little bit about already. So individuals with cystic fibrosis have 2 copies of the mutant allele, so they have no functioning copy of this chloride transporter, membrane transporter. And cystic fibrosis is common in a different population. So it's common among people of Central and Northern European ancestry. So Central and Northern Europe, that would be like Scandinavian countries, Germany, France, Great Britain. Those are Central and Northern Europeans. And approximately 1 in 25 people who have that ancestry are carriers of cystic fibrosis. Now that doesn't sound like a lot. That's just about 4%. But for a deleterious allele that without serious modern medical attention is 100% fatal in childhood, 4% of the population being carriers, that's really common. So what that means is that in the US, that if both parents are of European descent, about 1 in 3,500 babies will be born with cystic fibrosis. So for a genetic disorder, that's pretty common. If we look at African Americans, it's only 1 in 15,000, and for Asian Americans, only 1 in 32,000. That's more like the numbers that we would expect for a rare deleterious genetic disorder. So the question was then, is this some sort of heterozygote advantage? Well, it just so happens that the cystic fibrosis protein is the binding site for toxins of cholera and typhoid. So this has led to the hypothesis that the heterozygote advantage here is that if you're heterozygous for the cystic fibrosis allele, you would be more likely to survive a childhood bout of cholera or typhoid, both of which are bacterial infections. Cholera and typhoid are both fecal transmitted bacterial infections. Normally people get them by drinking contaminated water that's contaminated with fecal material, and that's currently a hypothesis. Now, some laboratory experiments to test this hypothesis have had mixed and not so fantastic results. So it's possible that this is the case, and it could be something else. But it's interesting that this protein is necessary for these bacterial toxins to work. And in both of these things, cholera and typhoid, they get into your intestinal tract, they're secreting their toxins, and what happens is they cause water to be excreted into the intestinal tract, and so you get severe watery diarrhea and you die of dehydration. So they both kill you essentially the same way. And the hypothesis is that if you were a heterozygote, then you wouldn't become as dehydrated as easily, and you would survive a round of cholera or typhoid, which many people do. Many people who in the historical times who had cholera or typhoid did get better and survive. Adults more likely than children. We have a vaccine against typhoid. We don't have a vaccine for cholera that I'm aware of, but mostly we've gotten rid of that by getting rid of fecal contaminated water, which is a good thing anyway. Another weirdly common recessive genetic disorder is Tay-Sachs disease, which I mentioned in an earlier lecture as well. So Tay-Sachs disease is also common in a few particular populations: Ashkenazi Jews, French Canadians, and because some French Canadians moved to Louisiana, those are the Cajuns in Louisiana. So those are populations, and there's a few other populations as well, but these are the major ones where this allele is common. And there could be a heterozygote advantage, because if we look at the amount of enzyme that's produced in the brain, carriers have half the amount of enzyme that's produced by this particular gene. So, but no heterozygote advantage has been found. People have looked for disease resistance because it's a gene that's normally expressed in the brain. People have looked for some kind of brain advantage, increased math ability, increased language ability in heterozygotes, and they haven't found anything. So it's possible that whatever the heterozygote advantage was here is for a disease that is now extinct. There are a lot of diseases that occurred during historic times that we have no idea what the organism or the virus that caused it was. We just have a description of the disease from historical documents, like, for example, the Great Sweating Sickness of Europe. We don't know what caused it. It was contagious. It was highly fatal. The saying was, fine at breakfast, dead by dinner. So you would get really sick with the Great Sweating Sickness. You would get a high fever. Of course, you'd sweat a lot. You would get really, really thirsty. People would beg for water, become delirious, and then die in one day. We have no idea what that disease was, but we know that there were epidemics of it that went back and forth across Europe. Is this possibly a disease like that one of these mystery diseases that came and went, and we have written records of them, but we have no idea what they actually were? Maybe. It's also possible that it's something called the founder effect. Sometimes we can see weird alleles common in particular populations because they were founded by a small group of individuals, and if there was a gene that was, or an allele that was common in that founding population, it might be common in this population. To me, that seems less likely, but that's another possibility for sure, because we don't know what the heterozygote advantage might be. Some deleterious alleles are dominant, which we already talked about polydactyly as an example of a deleterious dominant allele. Polydactyly is not that deleterious, though. Now we tend to surgically remove the extra pinkies and little toes that people have who have polydactyly. But even if we didn't do that, most of those people would not have too many negative consequences from that. But that's not true for all dominant alleles. Huntington's disease is also caused by a dominant allele. Huntington's disease is a disease that people, the average age of diagnosis is like in the 40s. And you get cognitive decline and also physical decline. So your brain deteriorates, and it's sort of like having Alzheimer's and ALS at the same time. People lose control over their bodies and lose the ability to walk, and then they also get dementia-like symptoms. So, and generally from the first symptoms until death is less than 5 years. And the age of onset, though, varies quite a bit. And it turns out that the gene that causes Huntington's mutates commonly. So new mutations for Huntington's aren't as rare as for other alleles. And if we look at the little pedigree down here, we can see typical inheritance pattern for a dominant trait. And you notice there's no carriers, because if a trait is dominant, we always see it in the phenotype. So if you have one allele for Huntington's, you will see it in your phenotype eventually, if you live long enough. This one, the gene for Huntington's, the gene that controls Huntington's disease, was one of the first genetic diseases for which the gene was found. And they did it thanks to finding a population where the Huntington's allele was very common. Because it's a rare disease generally in the human population, figuring out, even if you had a family with a couple of people with Huntington's, it would be difficult to figure out which gene is the one that's causing Huntington's, because what geneticists would then look for, they would compare the DNA of the different people, those with it and those without it, and try to figure out which genetic variation is always present in the Huntington's patients and is not present in the other people. And that can be difficult unless you have a population where there are lots of affected individuals. So they found such a population in an island, I believe it was off the coast of Venezuela, if I remember the story right. And they had the highest incidence of Huntington's in the world. And by studying that population in the 1980s, they were able to isolate the gene that causes Huntington's. One of the most famous sufferers of Huntington's was the folk singer Woody Guthrie. It wasn't realized that he died of Huntington's at the time, because symptoms of Huntington's can be mistaken for other diseases. So before there was the genetic test for Huntington's, people were often thought to have died of something else, like dementia or alcoholism or something, when they actually had Huntington's. So it turns out that Woody Guthrie's mother also had Huntington's, and he also passed it on to his daughters, also eventually developed Huntington's. One question that has been around since the Huntington's test became available as the first test really of a late onset genetic disorder, would you want to be tested? If you had a parent or a grandparent who was diagnosed with Huntington's, would you want to be tested and find out if you have the allele? Because if you have the allele, if you test positive, you would have a 100% chance that at some point in your life, you're going to develop the symptoms of Huntington's. So something else that they discovered when they were studying this population that had the highest incidence in the world of Huntington's is something happens there regularly that doesn't really happen in the rest of the world often enough to study, and that is that two people who both have the Huntington's allele have children together. So here, the Huntington's allele is the capital H because it's dominant, and the lowercase h is the normal brain protein that the Huntington's gene codes for. So if we have, one thing that they noticed when they were studying these populations is if both parents eventually came down with Huntington's and they looked at the children, if we just look at the Punnett square here, so capital H means you're going to have it, right? They expected that 75% of the children would have Huntington's and 25% would be normal. And in fact, what they found was that 2/3 of the children of couples such as this would have Huntington's and 1/3 would be normal. And the reason is because we don't have any of these individuals. And why do you think that is? Why do you think we don't have any of these individuals, are not known? And the reason is because being homozygous for the Huntington's allele we believe it's an embryonic lethal, because we just don't find those individuals. And in this rare population that has a high incidence of the Huntington's allele, we see 2/3 of children of couples that both have it as having Huntington's and 1/3 normal, which would make sense if this square doesn't exist. So it's probably an embryonic lethal. Another interesting condition is achondroplasia. This is the most common type of dwarfism. And if we look at a little pedigree down here, we can see something similar to what we saw for Huntington's, and that is that there's no carriers. So that tells us that this is a dominant trait. And we can also tell, because if we look, every time there's an affected child, there's an affected parent. Child, parent, child, parent, child, parent, child, parent. We don't see any unaffected parents having an affected child, and that would tell us that it was recessive, and then we would also have carriers. So this is dominant. Now, is this a sex-linked trait or an autosomal trait? So sex-linked traits, generally, that means things that are on the X chromosome, although we can also have sex-linked mutations that are on the Y chromosome. Either way, we would see affected individuals would more likely be boys, or more severely affected individuals would be boys if it's a sex-linked dominant trait. But in fact, it's pretty, in this little pedigree here, it's pretty equally male and female, so that tells us as well that this is an autosomal trait. And I have Peter Dinklage up here because he's probably the most famous in the world today sufferer of achondroplasia. And now the question of what happens to homozygous individuals for achondroplasia, I have looked, and I have not been able to find a scientific paper addressing this issue. There certainly have been many couples. This is the most common kind of dwarfism. There certainly have been couples where both parents had achondroplasia. If there have been any homozygous individuals, it's not been reported in the scientific literature one way or the other. I would suspect that it would also be an embryonic lethal because it is a dominant trait. Just like Huntington's is likely an embryonic lethal if it's in the homozygous condition. And I would suspect the same is true for achondroplasia. But I can't find any published scientific papers on that topic. Because it's an interesting one. Okay, so we talked about Mendelian inheritance patterns in this chapter. So we understand Mendel's results now based on how the chromosomes move during meiosis, his law of segregation, the fact that one of each of the homologous pairs of chromosome goes into each gamete. An independent assortment of the traits that he looked at is the result of independent assortment of the chromosomes during meiosis. And things that are called Mendelian traits have complete dominance and recessiveness. Non-Mendelian inheritance patterns, so traits that are non-Mendelian traits would be things that are polygenic, like height and IQ, and multifactorial traits that also have environmental components to them, to their phenotype. We talked about heterozygote advantage. So this is a case where an allele is deleterious when it's in the homozygous state, but it gives us some sort of an advantage in a particular environment to individuals who are carriers for that trait. So we talked about sickle cell anemia. If you're heterozygous for that, you have a selective advantage in the case of malaria, and for cystic fibrosis, probably a selective advantage in the face of cholera or typhoid. So Pedigree analysis we talked a bit about too, and we'll talk more about that in the next section, which is how we study human genetics, because we don't do experiments with humans anymore, and generally we also don't have any ability to get DNA from individuals who are dead. So pedigree analysis is how we can study inheritance. Genetic testing has become a big deal. You can order private genetic testing with 23andMe and other companies. And we should be cautious with these. It's pretty good for well understood alleles, but they're also giving people advice on diet and other things that really the science behind those specific alleles being associated with them is iffy. Also, often all we have are markers. They call them genetic markers that are associated with particular things, but we have no molecular mechanism. We have no cause and effect. The marker in question may not even be in an actual gene. It's just some sequence that's nearby, some gene of interest. So be cautious when interpreting medical information from genetic testing. Some is well understood, some not too much. Some is very, very speculative and is possibly giving incorrect incorrect information. Carrier screening is very, very useful. So if you have cystic fibrosis or sickle cell anemia runs in your family, it could be useful for you to get tested to see if you're a carrier. That's one of the very good and accurate uses for genetic testing. We'll talk a little bit about diagnosis, prenatal diagnosis. Newborn screening is good. There are some people who think that we should do a full DNA profile on all newborns, and that just raises all sorts of privacy issues, and what happens when that information is inevitably hacked and stolen from some computer somewhere. So to do prenatal testing, until recently, the only way to get a hold of fetal DNA was to get some amniotic fluid or a little piece of the placenta. So amniocentesis is sampling the amniotic fluid. This can't be done until around 16 weeks of gestation, so well into the second trimester. Chorionic villus sampling is taking a little piece of the placenta and through the cervix, and that can be done a little bit earlier. Neither one of these things is risk-free, however. They're both of these procedures are invasive procedures that can, in a low percentage chance, trigger a miscarriage or an infection. So, but we don't have to do that anymore. Now, fetal cells can be obtained from the mother's blood, which is really cool. So they just take a blood sample from the mother, which, they take giant blood samples from pregnant women and pretty much every time you go to the doctor to test for all sorts of things. And now the fetal cells can be separated from the mother cells and their DNA can be removed, and we can do we can answer questions about specific genetic disorders as well as looking for chromosomal abnormalities like Down syndrome. And this can be done earlier, and it's completely non-invasive and safe because taking a blood sample from the mother is not considered to be, I guess it's a tiny bit invasive, but they do it constantly during pregnancy anyway. And an interesting result of learning to do this is that it's been discovered that fetal cells, in fact, persist in the mother's body for decades after the pregnancy. Fetal cells, stem cells, settle into the mother's bone marrow and continue to divide and produce new blood cells for the rest of the woman's life, which is a very interesting finding. And we have no idea if this is beneficial, if this is deleterious, if it's neutral. We just don't know. This is a relatively new discovery, and it's not being studied by very many scientists. But why we would have these non-self cells being able to settle down into the bone marrow and start to produce new cells, and they're not being attacked by the body as invaders is really interesting. But it just shows how much the mother's immune system is modified to not react to the fetus during a pregnancy. So very interesting, but also means that we could get a lot more information prenatally. Another way that we can do screening is if in vitro fertilization is already being done. So in vitro means in glass. So this means that outside the body, fertilization has occurred. So this is they harvest eggs from the woman, and getting sperm from the man is much easier. And then they do fertilization outside the body and then implant the developing embryos. So here is a human embryo here. There's a little glass suction pipette over here, and another smaller pipette that's being used to withdraw a single cell. So they can remove one cell from the embryo, and that doesn't harm the embryo. It will continue to divide normally, and then they can do genetic analysis on that one cell by allowing it to divide. So this is available, but you wouldn't go through the huge expense of doing in vitro fertilization just to do this pre-implantation screening. But this would also mean that, if there was a particular genetic disease that ran in the family, you could not implant embryos that would be affected by that genetic disorder.